So I'm pleased to be able to introduce our next speaker, who undoubtedly doesn't need any introduction, but I'm going to do it anyway. Our next speaker is Kerry Ressler, and Kerry, of course, was the president of the Society of Biological Psychiatry last year. Last year, he got to give no presentations, scientific presentations at all, despite presiding over many things. Let me tell you a little bit about Kerry, who he currently occupies the Poitras Chair in Psychiatry at McLean Hospital. He's the chief of the Division of Depression and Anxiety Disorder at McLean, a relatively new chief, having been there probably 2, maybe 3 years. Kerry spent the bulk of the early part of his career at Emory University and at Grady Hospital. He was 18 years in total. He led the Grady Trauma Project, which is a project that many of us know a lot about and have been keeping our eyes on for many years, and hoping that you'll talk about a little bit today, Kerry. It's a study focused on understanding psychology, biology, and trauma-related factors contributing to PTSD and violence in over 10,000 participants in urban Atlanta. Like I said, last year he was the president of the society, and we enjoyed everything he did last year. One interesting thing about Kerry is that he was the first student of Dr. Linda Buck before she won her Nobel Prize, and Dr. Linda Buck was, of course, a UT Southwestern trainee and faculty member for a long time, so somebody that we keep track of. Linda Buck won the Nobel Prize in 2004. Kerry Ressler is a member of the National Academy of Medicine. He has over 250 publications, and his work focuses on translational research, bridging molecular biology and animal models with human genetic research on emotion. Kerry Ressler.
Thanks so much, Carol, and congratulations, Carol and Lori, on just a fantastic program this year. It's so exciting. Also, Carol, I can assure you that being past president is a much better gig than being current president. So. I'm going to tell you about some of the field's work today, and really it's my honor to be here today and to be able to share with you some of the progress in the field of post-traumatic stress. The field has grown so rapidly that for 30 minutes I decided I'd really focus on the process of memory consolidation and the unique components of that with PTSD. In terms of disclosures, I'm on the Scientific Advisory Board for Resilience Therapeutics and consulting agreements with Biogen. So the take-home messages that I'm going to talk about today is that post-traumatic stress is a tractable disorder. First of all, what you'll hear throughout is the shared neural circuitry of fear processing across all mammalian species. And that gives us a great deal of ability to understand the intermediate phenotypes of fear processing, and particularly trauma and fear consolidation. The thing that's perhaps most unique about PTSD in the psychiatric space is that we know when it starts. By definition, it starts at the time of trauma exposure. And that gives us enormous leveraging ability to both understand its mechanisms, but also to potentially be one of the few disorders in psychiatry that we could really prevent in a targeted fashion. And along those same lines, we often think of learning and memory disorders related to dementias. But PTSD, as I'll walk through all of my talk, is about learning and consolidation of memory processing. And we can really target that, and some of the greatest advances in neuroscience in the last two or three decades have been in the learning and memory space. So I'm going to start with an overview of PTSD and fear, introduce you to the concept of memory consolidation and some of what's been known about it. Then I'm going to present several stories, as Carol mentioned, from our Grady Trauma Project, but particularly our emergency department work, showing human evidence of memory consolidation in the processes using neuroimaging, physiology, and some molecular approaches. And then switch over translationally to understanding the process of fear consolidation in mice at a behavioral circuit and molecular levels. And all of this is towards the goal of, if we can understand the biology of memory consolidation, we could potentially have the same kind of approaches in our emergency departments to prevent PTSD as we do for heart attacks or strokes or other things. If we knew who was going to develop it or who wasn't, and we had the right pharmacological or therapeutic approach, one could potentially prevent its development. So this is a shout out, first of all, to David Ross in the National Neuroscience Curriculum Initiative, really just a fantastic educational program that's been seeded in biological psychiatry and is really focused on many components of training neuroscience to young psychiatry residents and psychiatrists. And this great review that David put together really explains sort of the framework of PTSD, starting with the circuits. So the amygdala is in many ways the final common pathway of fear processing, and I'll talk a lot about consolidation processes in the amygdala, but it's always being modulated by the medial prefrontal cortex and the hippocampus. And basic Pavlovian conditioning, the idea is that there's a conditioned stimulus that's previously a neutral stimulus, in this case maybe driving down the street or a car or a specific part of town or something, but that is paired with an unconditioned stimulus, the horrible thing that happens in the trauma, the aversive event. And that one trial learning, we evolved for one trial learning of dangerous events, in part through a whole system of circuits, and I'll talk about the system consolidation component, but also ending within the amygdala component, so that in the future, that trauma cue reminds and reactivates the fear system as if one is reliving fear and reliving the trauma all over again. And again, we know it's modulated by these areas, leading to multiple downstream hardwired processes, and I often like to say that this is one of the few places in our field where we know specific hardwired connections from the amygdala to subcortical and brainstem areas leads to a whole set of symptoms that we can observe very clearly in a panic or fear response. The heart rate changes, the respiratory changes, the GI distress, the sweating, etc. And that this memory process starts initially with short-term memory in the very minutes and hours of the event. But that's relatively quickly consolidated, and this is what will be the theme of my talk today, into long-term memory. There's been a lot of excitement over the last decade or so on the idea of reconsolidation, started with Karim Nader and Joe Ledoux, that maybe being able to reactivate a memory makes it labile again, though the constraints on this in humans remain to be seen, how much we'll be able to manipulate it. And then extinction, being able to learn new safety components, we think is very much the process that goes on with prolonged exposure, the primary therapy in most of our anxiety disorders. So these are all different components as well as different aspects of cognition, generalization, discrimination, sensitization that occur in PTSD. But again, today I'm just going to focus on what we know about consolidation. So Jim McGaugh, in some ways, is one of the leaders of the concept of modern consolidation. This is a great review he wrote almost 18 years ago now. And the basic idea, again, is that over time, short-term memory is transmitted to long-term memory, and work from his group in particular on the role of adrenergic systems, the beta-adrenergic pathways, and the cortisol systems with stress, and how those mediate hippocampal and amygdala consolidation were foundational for a lot of current ideas about stress-related consolidation. And what he and others showed is that memory consolidation at a cellular and systems level, again, is made up of a short-term process, then mediated more long-term, and then with long-lasting memories. And with work by Eric Kandel and many others in the early 80s and 90s, focusing on the molecular mechanisms of the most simple forms of vertebrate memory or invertebrate memory, showed that you could identify the molecules in part for some of these simple systems involved in the early short-term memory phase, the immediate early genes, as well as the longer-term genes that turn on that lead to the structural changes mediating memory. In the field of fear, Joe Ledoux, as well as his parallels in the field, Mike Fanzelo and Mike Davis and their offspring, really over the last 2 or 3 decades worked out what the amygdala circuitry is related to fear. And some of Joe's work was the first to show that that the amygdala mediates LTP, like long-term potentiation type processes following fear conditioning, so that you get increased excitatory potentials in slice physiology or in vivo after paired fear conditioning compared to control animals. And that can be shown multiple different ways, both in terms of slope and amplitude. And we now know that the LTP in the amygdala and the basic fear consolidation process is NMDA dependent, calcium dependent, BDNF dependent, dependent on most of the processes that in cell culture have been shown, or slice have been shown to mediate LTP, also mediate the basic consolidation mechanisms of fear memory in the hours after trauma. My own work in this area started with Mike Davis in the late 90s, early 2000s. And what had not happened yet was a molecular biology of amygdala function. And so Mike and I began looking at a number of gene pathways, and we didn't have the sequencing and other approaches that we have now. So I cloned about 40 different genes that were known to be involved in LTP-based molecular mechanisms in slice physiology, and we did in situ hybridization to see how those changed over the course of the consolidation window. And so several take-home points. First of all, we were delighted to see that across the amygdala and other areas I'll talk about in a second, that we could at the whole brain regional level and the cellular level see these same immediate early peaks, longer term peaks, et cetera, as was predicted from aplesia. Secondly, we saw a set of genes that were downregulated instead of upregulated on a similar time course, which is quite interesting. And for example, Geoffrin is involved in GABA modulation, and downregulation of Geoffrin in the transient consolidation period leads to hyperexcitability because of decreased GABA function. Another point is we were able to see a lot of these changes in the amygdala as we expected. So this slide just shows a control with seizures, showing immediate early genes and others, control animals, and then fear-conditioned animals a few hours after fear conditioning, and again seeing much activation in the amygdala temporal lobe areas. But also importantly, we didn't just see it in the amygdala. We talk about in the field the amygdala as the final common pathway, but we saw plasticity at almost every part of the brain. Certainly every part of the brain involved in this pathway, from the sensory systems through the thalamic pathways, hippocampal pathways, and brainstem pathways. And this just shows after tone fear conditioning, seeing CFOS as an immediate early gene. Unpaired, we see no signal. With paired tone shock learning, you see a big pulse of CFOS that then goes down over time. Neura involved, most known for dopamine, is also involved in plasticity in a number of ways, had a slightly different time course. BDNF looks more like this prolonged time course, et cetera. So it suggests that there's a whole set of molecular pathways that are activated with different time courses throughout the whole brain circuit, mediating the tone representation. So I want to now shift gears and tell you about some of the human data implicating amygdala and hippocampus in the consolidation process, and then I'll switch back into more of the mechanistic components. We got involved in this work with a collaboration with Charlie Nimmeroff at Miami and at Emory and Grady Trauma Project, where we started working with the emergency department at Grady, which is one of the largest tertiary care centers and one of the primary trauma receiving facilities in Georgia. And we were able to get bloods added to the stat pack. So as somebody would roll off of the ambulance and they'd get bloods for crossing and typing and other things, we could get experimental bloods added as well. And then we were able to, and then if they didn't consent, the bloods were thrown away. If they did consent, we would interview them when they were medically stable. So it basically allowed us to get peripheral biomarkers in the consolidation window within the first couple hours of the trauma, and then we'd follow them up for multiple time points afterwards. And some of this work has led now to, so that was about 500 people, and now we're part of a large multi-site consortium funded by NIMH with the direction of Susan Borgia and Ferris Tuma, with the PI of Sam McLean at UNC, Kirsten Conan and Ron Kessler and myself, and many, many collaborators. And the idea here is to have at least 5,000 completers and maybe quite a bit more depending on the process, that we can really work out the entire timeline at a biomarker level, a physiological level, digital phenotyping level, et cetera, in the aftermath of trauma. But that's just beginning. So some of the data that's come out of the first study is data consistent with the idea that the amygdala and cingulate areas that we know are involved in chronic PTSD are also predictive of PTSD in the aftermath of trauma. So this work, the imaging work led by Jenny Stevens and the whole emergency department crew, this is the trajectory of PTSD symptoms from enrollment over the 12 months. And we imaged people with structural, functional, and task-based imaging at 1 month. And what Jenny was able to show is that their amygdala activation at the time of 1 month predicted their PTSD symptoms at 3 and 6 months later. Sana Van Rooij worked on similar population, focusing on the role of hippocampus in inhibiting the amygdala. And what she showed was that hippocampal activity that we normally see in a response inhibition, like go-no-go task, the hippocampus activity during this response inhibition task was inversely correlated with later PTSD. So basically, the more the hippocampus could inhibit could be activated during response inhibition, the less likely one was to have PTSD. The less the hippocampus was doing this generalization, discrimination, response inhibition task, the more likely one would be to later develop PTSD. And that held up with a number of important covariates as predictors using elastic net regression. So some of the most exciting current data that we're working on is led by Becky Heinrichs and Tanya Ivanovich. And this is looking at physiology in the emergency department. We know anecdotally that peritraumatic dissociation and other measures of self-reported stress are critical indicators of later risk. But we haven't had as a field a good quantitative metric in the immediate aftermath of trauma. And what Becky did was using an easily portable galvanic skin device called eSense to measure the skin conductance in the emergency department as people were telling us about their trauma in that first few hours. And then she looked at PTSD symptoms 3, 6, and 12 months later, and we found a remarkable correlation in their immediate sympathetic arousal state at the time of the trauma, predicting later PTSD, and that's on both a continuous and a categorical level of analysis. We're just beginning some of the analysis at the molecular level, but this is a paper from Ari Shatlov's group from about a decade ago, where they were doing work in Israel, and they found a number of genes that were differentially expressed in the immediate aftermath of trauma that would predict later PTSD. And two main take-home points here. One is most of the paths that they found differentially expressed were involved in RNA processing, and I'll tell you one example of a new story from that in a minute. And then secondarily, one of the genes they found differentially expressed has been one of our long-term favorites, FKBP5, which is involved in differential risk for PTSD in the aftermath of trauma, and it's been associated with amygdala activation, hippocampal function, hippocampal structure, and hippocampal connectivity. And is very much involved in the stress response axis and likely tied in both the HPA feedback system and the sympathetic arousal processes we see with the galvanic skin response. One example from our new data on the RNA expression in the aftermath of trauma, we haven't found a main effect yet of mRNA, but at the non-coding RNA, Guia Jafantes led a story that found a non-coding RNA called Gas5 that's differentially expressed in those who go on to develop PTSD versus not at both 1 and 3 months. But again, the blood draw is in the immediate aftermath of trauma. And Gas5 is very interesting because it's a non-coding RNA that actually mimics the glucocorticoid receptor binding site of cortisol. So it's a non-coding RNA that functionally interacts with GR. So both FKBP5 and Gas5 are both interacting through GR regulation in the immediate aftermath of trauma and likely very much tied to this sympathetic arousal and HPA axis arousal process that we see. And polymorphisms that are expression-linked QTLs, EQTLs, that predict Gas5 levels are also differentially associated with amygdala activation. So I wasn't going to talk about genetics, but I thought I'd have one slide because the field is so exciting right now where genetics are. And this, what's happened in the last 5 years really, is that in the same way that the GWAS fields have really exploded for schizophrenia, depression, and other areas, they have as well for PTSD that we were a little bit later starting. And the Psychiatric Genomic Consortium and the Million Veterans programs are two of the largest and most successful so far. The Psychiatric Genomic Consortium group, led by Kirsten Conan, Israel Liebrazon, Caroline Nevergelt, and many others, showed this was the first freeze data confirming heritability and several interesting points in our freeze 2 that's currently being written up has a number of GWAS hits. And Murray Steen and Joe Galertner have recently presented data that the Million Veteran programs, with over 100,000 samples total, now has many really nice hits on the Manhattan plot, really suggesting that the field has hit the inflection point, and that as these data are combined and other data are coming in, there are going to be more and more genome-wide level strong hits supportive of understanding PTSD. This one was particularly focused on the hyperarousal symptoms. One of the interesting findings here, though, is that one of the main hits was the CRH locus, corticotropin-releasing hormone receptor, which has been one of the biological outcome pathways of the HPA stress axis for a long time. So that's very exciting, and I'll come back to that in 1 minute. So I want to shift gears and talk about how we understand amygdala consolidation in animals. So again, the amygdala is very conserved across species, and looking at basic Pavlovian learning and memory processes where we pair, for example, a tone or an odor with a foot shock, we can understand the molecular mechanisms of amygdala function. And coming back to the CRF just for a second, so I showed you that one of the top hits now from the Million Veteran Program includes a locus that is over the CRH receptor, and some of these SNPs are EQTLs for CRH receptor. The field has done a lot of work over the years, and this is just one example of either overexpressing CRH using virus in a mouse amygdala, or making CRF cells overactive by knocking out GABA in those cells. Both leads to increased activation, increased anxiety-like behaviors, and importantly, in the aftermath of fear acquisition, the CRF cells that are hyperactive because of GABA knockout show significantly increased fear consolidation. So I want to shift gears in talking about the animal models to some of the structural changes that we know occur with consolidation. This is built on a whole literature of sensory systems changes with learning by Mike Merznick, John Cass, Mike Stryker, and many others. Also in the PTSD field, there's been a number of interesting human studies suggesting somatosensory changes at a structural level with early trauma. So the first example I'll show you is auditory cortex after tone shock fear conditioning. Orion Kiefer showed he did several days of tone shock fear conditioning and then waited 3 weeks. And when he did that, he then looked first with voxel-based morphometry ex vivo imaging and found significant increased density, gray matter density, in a number of brain areas that are associated with the fear processing pathways, the auditory cortex, parts of the amygdala, and the insula. But we were able to use fluorescent genetic indicators to then look at spines, and he was able to show a rightward shift in spine density in a shoal analysis in that animals who had auditory fear conditioning had significantly more spines and higher spine density and more mature spines than did animals that did not have fear conditioning, suggesting that part of this whole process is associated with structural changes in the auditory cortex. Nina Banerjee went on, in collaboration with Robert Liu's lab, just to look at some of the mechanisms of this. And one thing she was particularly interested in was perineuronal nets that have historically been primarily associated with development. But and we know that perineuronal nets primarily surround GABAergic cells in cortex. But she observed that these appear to be very dynamic in adult animals following fear conditioning, such that there's a significant increase in the perineuronal nets, both in the density of the WFA stain as well as the number of cells surrounded by them in a transient fashion during consolidation. She did a number of studies, but just one here to show mechanistically and causally. She showed that if she ablated the perineuronal nets with chondroitinase ABC right before or immediately after fear conditioning, so this is the perineuronal nets at baseline, and this is those after they've been ablated, and then looked at consolidation, she had a robust inhibition of memory consolidation, suggesting that the perineuronal nets were somehow, that dynamic increase in them was somehow causally related to the auditory cortex-based consolidation. And the current model is that the perineuronal nets, when they surround the parvalbumin cells, actually decrease the excitatory inputs onto these cells. And so that by ablating them, the transient increase in them would effectively disinhibit their activity on the pyramidal neurons in a transient way. And again, a theme coming throughout here is whether it's transient GABA downregulation, transient perineuronal net increase, transient HPA increase. These are all things that together, both in cell slice physiology and in the amygdala, are consistent with enhancing the synaptic plasticity period during consolidation. Carol mentioned our early work in the olfactory system, and we've always had some level of olfactory work ongoing just because the tools are so amazing. And the take-home message of what we now know about the olfactory system is there's about 1,000 odorant receptor genes in the nose, but they all project, each cell only has one receptor type, and it projects specifically to a specific glomerulus in the bulb. And we found with Seth Jones a number of years ago that if you do olfactory fear conditioning, so 3 days of 10 odor shock pairings and wait a few weeks, you see a robust increase in the number of neurons in the nose and the density of that glomerulus. So these are specific neurons that recognize one particular odorant in the nose, and this is at baseline. After these animals have been trained to be afraid of that odorant, you wait a couple weeks, you get this robust increased density of the axons into the bulb, and that's mediated by just more neurons in the nose. And I'm not going to go into it, but Philomene Morrison has data that this mechanism is TrkB and BDNF dependent, that increases the cell survival of these axons. Philomene also recently showed both replicated this main effect, both in terms of the neurons in the epithelium of the nose and their axons in the bulb, but very interestingly showed that if you then later on extinguish these animals so that they no longer care about that odor, the number of neurons in the nose and bulb axons diminish back to baseline. So it seems to be a dynamic structural process in sensitivity to the odor that the animal is afraid to. So the take-home message of the sensory system plasticity is that it seems that when you become afraid of an odor or a tone or other cue, the whole system, the sensory system, has increased sensitivity. We don't think that the olfactory bulb or the auditory cortex knows that this is a fearful thing, but it is more sensitive to that for future arousal. I talked about the non-coding RNA changes that we see in the human periphery in the aftermath of trauma. Brian Diaz, who did a lot of the olfactory work, also identified microRNA that were involved in fear consolidation. He did a microRNA array at this point before RNA-seq, and found a couple of microRNAs, one of which robustly replicated microRNA 34A that was increased in fear consolidation. He showed it was predicted electronically to target the Notch pathway, historically involved in development and less known about its role in synaptic plasticity, showed that this microRNA experimentally targets Notch, and also found that naturally a number of Notch ligands, JAG1 and disheveled, as well as the receptor for Notch1, were all dynamically regulated in the aftermath of fear consolidation. He did a whole series of studies focusing on the mechanisms of Notch-mediated plasticity, but one of them here shows a virus that he made that effectively an antisense, a sponge for the microRNA 34A, putting it in the amygdala, showing that when you do that, you have no effect on acquisition, but a robust decrease in memory consolidation, fear consolidation. This and many other studies led to the findings that Notch pathway, in part regulated by microRNA 34A, is a critical mediator of synaptic stabilization. And that normally at baseline, you have more Notch signaling, which leads to synaptic stability and impairs consolidation. But in the transient period after fear learning, you have decreased Notch signaling through increased microRNA 34A, which leads to transient synaptic instability and plasticity and enhanced consolidation. I don't have time to go into it, but we and others had a whole series of studies as well focusing on beta-catenin. And beta-catenin we know interacts with Notch as well. And the take-home message was similar, that beta-catenin is transiently decreased through phosphorylation in the aftermath of trauma or fear learning, allowing spines to basically split and then restabilize. So that a number of genes and pathways that we previously know were involved in development are also involved in synaptic stabilization and transient synaptic plasticity in the aftermath of trauma. The field's moving more and more towards the cell types in the amygdala. We now know there are probably tens, if not hundreds, of different kinds of cell types in the amygdala, and how can we understand their function and therefore how they're related to fear consolidation. One particular cell type that we've been focusing on that's marked by the Phi1 cells within the basolateral amygdala seems to inhibit fear or be a fear-off pathway. When you activate these cells with optogenetics, you get basically a blockade of the flow-through physiologically through the amygdala. And importantly, when you activate the cells in vivo with optogenetics, you block fear consolidation, or if you inhibit the cells with halorhodopsin, you enhance fear consolidation. So separate from the molecular question, at the microcellular and circuit level, you can also block consolidation or enhance consolidation by turning these pathways on or off. And specific drugs that activate those cells also affect consolidation, in this case, block consolidation in a predicted way. So, and the final story I'll tell is tachykinin 2. That is a neuropeptide that marks another important cell population. Raul Andero, who's now in Barcelona, did RNA-seq in animals at home cage versus after fear and during the consolidation of fear conditioning. And one of the genes that he found differentially regulated dynamically is TAC2, like we see here. What was cool about this finding is not only was the peptide differentially regulated, it marked a really important cell population. Before this, the centromedial population, the output cells of the central amygdala, we didn't have a known cell marker of a subtype, and it turns out that these mark the centromedial population very nicely. And Raoul then went on to show that pharmacological blockade of this pathway impairs consolidation. So he gave a drug, asanatan, which was known to block the TAC2 receptor, the NKB receptor. Gave it either before fear conditioning, 10 minutes, 1 hour, or 4 hours, or 24 hours after fear training. And he again found a nice consolidation window that if you blocked the receptor before conditioning or in the first 30 minutes or a few hours afterwards, you would get a decrease in consolidation. But if you waited till 4 hours, you did not. He also showed that activation of the PACAP receptor, which we know is associated with PTSD and with fear learning, and normally happens during fear consolidation in the central amygdala, that's blocked or normalized with asanatan blocking TAC2. He went on to show that in the amygdala, these drugs work directly. So he did acquisition through a cannula specifically, then immediately after acquisition, injected the drug that blocks the TAC2 receptor and saw a robust decrease in fear consolidation the next day. And he was able to push it in the opposite direction by overexpressing TAC2 in the amygdala with the virus. And when he did that, he found an increase in consolidation. So again, we initially found TAC2 as increased transiently at the mRNA level, replicated it with protein, and showed we could push it up or down by knocking it out or inhibiting it pharmacologically or overexpressing it. And finally, if you combine overexpression with asanatan, he replicated the increased consolidation and then normalized that increased consolidation with the antagonist asanatan. So I've walked you through a number of stories relatively rapidly, but the theme has been the same, that we can understand the concept of memory consolidation broadly, and that's particularly relevant to fear processing and trauma processing. And that trauma memories don't become permanent immediately, rather they become consolidated over the course of hours to days. And that if we can better understand the neural circuits and thus the molecular pathways that mediate this consolidation, we may be able to create a whole new set of approaches in a rationally designed way to block fear memory formation, trauma memory formation in the aftermath of trauma. And that this consolidation happens at the level of amygdala and hippocampus, but it happens all the way out to the level of the sensory systems, and I showed you the data from auditory and olfactory systems, but as well as at the cellular level throughout, including different cell pathways within the amygdala. And it suggests a number of hypotheses that we could, by impairing molecular consolidation through HPA axis function, and some work by Zohar and others have focused on cortisol pathways, propranolol, but I think it's fair to say we don't have any of the right tools yet that we can safely say this is going to work. So we need more efforts. And a number of new pathways, BDNF track B pathways, beta-catenin and Notch pathways, and a host of microRNA and other epigenetics targets potentially available. Understanding cellular consolidation gives us new targets, as I showed with the CRF cells, the Thy1 and the TAC2 cells. And there's also the concept of systems consolidation. If we did, for example, TMS to activate or inactivate the right dorsolateral circuit or medial prefrontal circuit in the immediate aftermath of trauma, would that be an opportunity? Are certain kinds of talk therapy an opportunity? And things like xenon and other agents that seem to block NMDA function transiently, maybe even ketamine, used in the right way with the right timing, might also block consolidation in the immediate aftermath of trauma. I'm really just the cheerleader of a great team. I've tried to acknowledge them as I've went, and I thank you very much for your attention.
Thanks, Kerry, for a terrific talk. I'll ask the first question. Many years ago, in working with the Institute of Medicine and looking at war trauma, we noticed that the biggest predictor of PTSD, of no PTSD, was previous war experience. Why should anticipation, I guess, or previous experience modulate the whole process that you've been describing?
Yeah, I think there's a lot of ways that we think about amygdala sensitization. I think, for one example, we know that childhood trauma is the biggest risk factor for most things in psychiatry. And we know that childhood trauma sensitizes the amygdala, that you can see at both the functional and a structural level. And I think that's going to be the case for other kinds of traumas as well. But it's also the case that all trauma difficult experiences are not always bad. For some people, they become resilience forming or there's traumatic growth there. So I think different past experience will definitely mediate how new traumas are experienced, but there's going to be individual variation in how that's used.
If there's no other questions,
Thank
would you join me in thanking Kerry Ressler?